Scott Brown's Otology, Chapter 47, Anatomy of the Cochlea and Vestibular System relating ultrastructure to function. General characteristics of supporting cells. General characteristics of supporting cells. The supporting cells provide mechanical support to the epithelium and thus to the hair cells. Their cell bodies contact each other and rest on the basement membrane that underlies the sensory epithelium. Rod-like phalangeal processes extend from the cell body, interdigitating between the hair cells to the luminal surface, where they expand to fill the spaces between hair cells. Supporting cells possesses a fairly extensive cytoskeletal system that is particularly well developed in the cells of the organ of Corti. These phalanges contain bundles of microtubules which can act like scaffolding poles or suspension struts to provide structural support described in more detail later. At the apical luminal poles, there are cytoskeletal assemblies that contact actin arranged in filamentous bundles running parallel to the luminal surface of the cell, anchoring to the adherence-like regions within the intercellular junction that joins to the adjacent hair cell. Actin bands that run circumferentially around the neck or cortex of the epithelial cells are present in all epithelial cells and are thought to provide structural support at the points of adhesion between adjacent cells. These cortical actin bands are unusually wide in sensory epithelia of the inner ear, indicating the importance of rigidity at the apical surface of these tissues. Supporting cells are functionally coupled to each other by substantial numbers of large gap junctions, and gap junctions are sites of direct cytoplasmic communication between adjacent cells where clusters of pore-forming channels, proteins in the membrane of one cell and are in direct register with clusters of pore-forming channel proteins in the membrane of its neighbor. When these clusters of protein dock together, they can form continuous aqueous pores that connect the cytoplasm of the adjacent cells. The protein subunits that form gap junction channels are members of the connexin protein family. At least 20 different types or isoforms of connexin have been identified. 6 connexins form a hemichannel and or connexon, and the connexons of two adjacent cells align symmetrically to form the communication pathway between the cells. Gap junction channels are clusters in the plane of the membrane to form plaques that can contain up to several thousand connexins. The channels allow the passage of small metabolites up to 1.2 k Dalton in size, ions, and second messengers, coupling the cells both electrically and chemically. Numerous gap junctions are present at points of contact between adjacent supporting cell bodies and between the head regions of adjacent cells, but there are no gap junctions associated with hair cells. The large size and number of gap junction plaques between all supporting cells mean that the supporting cell population can be regarded as a functional syncytium from which hair cells are functionally isolated. In all vertebrate classes from fish to mammals, gap junction plaques in inner ear sensory tissues are typically enormous, among the largest in the whole body, covering several square micrometers (meter square) in area and containing several thousand channels. One suspected role for supporting cells is thought to be the removal of excess potassium ions from the intracellular spaces of the sensory epithelium during hair cell repolarization events. Such a mechanism could thereby maintain the low potassium environment around the body of the hair cell necessary for transduction and sensitivity to stimulation. It has been proposed that the gap junctions provide a means to ferry the potassium away, preventing local accumulation. Gap junctions in the organ of Corti and in mammalian vestibular sensory epithelia contain two connexin isoforms, CX26 and CX30. Mutations in the genes for these connexins have been identified as causes of hereditary sensorineural hearing loss. These mutations in the gene encoding for CX26 GJB2 are the most common cause of non-syndromic hereditary deafness. There is increasing evidence that the gap junctions with a particular connexin composition, in particular CX26, are important during development of organ of Corti. However, connexin mutations appear to have much less effect on the vestibular system. Thank you.